Hi everyone. In today's video, I just wanted to go through the different types of active transport and explain to you what active transport actually is. So active transport is when a substance is too large to pass through a channel, is incapable of dissolving in the lipid bilayer, and has to move up a concentration gradient. This last stage that I mentioned is probably the most important, as active transport almost always involves a substance moving from lower concentration to higher concentration, essentially moving up its concentration gradient. So in this case, in order for this process to occur, we can visualize it with the analogy of someone requiring a ball to move up a hill. Now, as I mentioned in my previous video about passive transport, if the ball was present at the top of the hill, we could simply let the ball roll down it without any energy being required. However, in this case, as the ball has to roll up the hill, we require some energy in order to push it upwards. This is when the cell comes into play and is able to provide energy in terms of metabolic energy in order to allow for the substances to move across a particular membrane. This energy is provided by ATP, adenosine triphosphate, and involves usually a carrier protein that specifically and reversibly binds to the molecule to facilitate the transport. So if we have our plasma membrane here, and a molecule wants to pass through it, then if we have, for example, a particular method of transporting the molecule that requires ATP, then once ATP binds to this site, the molecule will be able to pass through. This here, energy that is provided directly by the hydrolysis of ATP, is actually called primary active transport. But we'll get back to that later. Firstly, I just wanted to go through the different types of active transport that exist within cells. Firstly, the two main headings under which active transport can form are endocytosis and exocytosis. There are four main types of endocytosis that can occur within cells. Firstly, there is pinocytosis, phagocytosis, receptor-mediated endocytosis, and finally, transcytosis. In terms of exocytosis, there are two main types. Firstly, there is vesicle-mediated trafficking, or vesicle-mediated exocytosis. And like endocytosis, there is also transcytosis. Now, before we get into these, I'd like to go back to the concept of primary active transport, as many of these transport processes fall under this umbrella. So, what is primary active transport? Well, in a nutshell, it is essentially the energy for the transport coming directly, as I mentioned earlier, from the hydrolysis of ATP. And I think the best way to explain this is by looking at an example. So say if we have the lipid bilayer present here, the most clear example I would say is the sodium and potassium pump that exists within many cells. So this pump if it is open to the inside of the cell, will look something like this. And essentially, if sodium molecules are present within the cell, then this molecule contains 3 sites for the sodium molecules to bind to in order to be transported out of the cell. And now you may be wondering, how does the transporter get the energy to do this? Well, it also contains a certain site where a phosphate group of an ATP molecule is able to bind. And this provides the energy from the bonds that are present in the ATP molecule to cause for a change in the conformation of the particular transporter protein to therefore allow for the sodium molecules to pass out of the cell. Now, even though the sodium molecules have been passed out of the cell, the ATP that was once bound to the particular part of the transporter, or the phosphate group, stays bound to it. And as you may know, on the outside of the cell, there are many potassium molecules present. And these potassium molecules are able to bind to their sites, which there are 2 on a particular sodium-potassium transporter. And the binding of these, in combination with a particular phosphate group, and eventually this phosphate group is removed, allows for another conformational change, wherein the transporter moves back to its original state, and the phosphate groups, the potassium groups that were present on the outside of the cell can now be present on the inside of the cell. So essentially, this protein is able to flip and flop in order to open and close. This is all through the direct hydrolysis of ATP. And therefore, these molecules are able to move against their concentration gradient. And this particular transporter is able to regulate ion concentration in and out of the cell. So in a nutshell, primary active transport involves the hydrolysis of ATP, and also this phosphorylation of a particular transport protein causes for a change in its shape and therefore the pumping of the bound solute across the membrane. So once again, originally Na molecules are able to bound to their particular sites. A phosphate group is able to attach to provide energy for this process to occur. Then the transporter changes shape, in which the Na molecules are able to flow out and potassium molecules are able to bind to their sites. The phosphate group is removed and may be added to another ADP molecule in order to form ATP. And then it is, because the phosphate group has been removed, the protein transporter is then able to return to its original shape, which was present in the first image here, and therefore allow for the process of the potassium molecules to pass into the cell. Now that's the process of primary active transport. There is also secondary active transport that is present. Now, secondary active transport is a little bit more complicated than primary active transport, in that it is actually driven by the energy stored in ionic gradients created by the primary active transport pumps. So in this case, actually, two substances are able to move through the membrane at once, in which there is a coupled system. So there are two types of coupled systems that can occur within a cell. If we have, say for example, the lipid bilayer, and a particular transporter is able to allow for, through the direct hydrolysis of ATP, the passage of sodium into the cell, therefore moving up its transportation gradient, concentration gradient. The establishment of a concentration gradient in the opposite direction is able to allow for another molecule to move out of the cell. And this is a secondary active transport process. As these molecules are moving in the opposite direction, this is actually called an antiport system. On the other hand, if the molecule was moving in the same direction as where the sodium molecule moved, then this system would actually be called a symport system. Therefore, this is seen also in a sodium-potassium ion pump as we mentioned earlier, the concentration gradient that may be formed by this particular pump may establish another ionic gradient which can allow for the transportation of another molecule. So in a nutshell, secondary active transport involves two different gradients. One, which is formed by the ionic gradient, which allows for the transportation of a secondary molecule, and the other being the forced gradient, essentially, that is formed by the direct hydrolysis of ATP. And this essentially is what allows for the formation of this ionic gradient. Now, there's a type of transport that I would like to also mention, which is quite important in cells as well, and is a concept that relates to all other types of transport, and this is called bulk transport. Now, bulk transport is quite an important concept because it essentially involves the transport of solutes against their concentration gradient and requires energy and involves the bulk movement, so many molecules, across a membrane. And the two types of bulk transport are endocytosis and exocytosis. And the reason why I stress this type of transport quite hard is because it is essentially the basis for all types of primary and secondary active transport processes. So let's take a deeper look at what endocytosis is. As I mentioned before, there are 4 different types of endocytosis. And firstly, we will be looking at phagocytosis. So, what is phagocytosis? If you would have seen my video on the different types of white blood cells, you would have seen that phagocytosis is quite an important mechanism of action for these cells. It essentially is when a cell engulfs another substance, in this case a particle, and it essentially forms a sac around the particle, and this sac is called a phagosome. And it can also link with other organelles within the cell in order to have a specific action on the particular particle. This may be, for example, with a lysosome, if the particle has to be broken down. So, phagocytosis. This process is seen in, for example, neutrophils. So these neutrophils, if I were to roughly draw one here, is able to consume a particular pathogen in a Pac-Man style manner. So the pathogen is present here, and essentially the pathogen is able to pass into the cell in a vesicle in order to essentially be destroyed. Next, we will look at pinocytosis. Now, this process is quite similar actually to phagocytosis. It essentially is identical. Instead of particles being present, however, there is droplets of liquid. So, in the same manner, droplets of liquid are transported into the cell through protein-coated vesicles. In this case, similar to phagocytosis, receptors are not required. So for example, if we have a particular cell here, in a similar Pac-Man-like manner, it is able to consume a liquid and therefore, through a vesicle, is able to allow for the transportation of that liquid within the cell and therefore allow it to interact in particular manners. The next type of endocytosis that I'd like to go through is receptor-mediated endocytosis. This receptor-mediated endocytosis is seen quite often in cells, as it's seen in many pharmaceutical drugs and also other hormones. It's when particles entering the cell can bind to specific proteins at specific parts of the membrane and therefore can be brought in through vesicles. So for example, if we have a particular membrane of a cell present here, then a receptor is present here. A particle is able to come and interact by having a complementary type to that receptor and essentially be brought into the cell through a vesicle by the interaction between these receptor and particle, and therefore be brought into the cell. And similar to all other types of endocytosis, this vesicle and the subsequent particle inside it can interact with various organelles. Now, before I actually talk about transcytosis, as you know, it's a process that is present in both endocytosis and exocytosis, I would like to first talk about vesicle-mediated exocytosis. This process is similar to endocytosis because it involves vesicles. So, vesicle-mediated trafficking. This is when essentially a vesicle is able to transport a substance out of the cell by fusing with the plasma membrane and therefore not allowing anything else to be transported in or out. Similar to the process that I've outlined here, there is some sort of a receptor present on the lipid bilayer. So if this is the lipid bilayer of the cell, and this is the inside, there is a particular receptor that is present, and the vesicle that contains the particular substance that has to be taken out through exocytosis contains complementary receptors on itself in order to allow for the fusion of the vesicle with the plasma membrane and this fusion essentially allows for the substance to be taken out of the cell. And the fusion is quite important, as even though our intention is to move a substance outside of the cell, we don't want anything else to move in or out. And therefore, this particular process is important, as after the substance has been moved out of the cell, so this is the particular substance, the plasma membrane can actually return to its normal shape, as if nothing had occurred. The final method of active transport that I'd like to go through today is transcytosis, and this is a process that occurs with a combination of endocytosis and exocytosis. Transcytosis is essentially when a vesicle has been brought into the cell through receptor-mediated endocytosis and can therefore fuse with the plasma membrane to deliver its contents into the extracellular matrix once again for another cell to pick up. So essentially, it's when, if we have a cell present here with a nucleus, a substance is able to move into the cell through a vesicle, move through the cell, and then pass out of the cell. Once again, here through receptor-mediated endocytosis, and then pass out through vesicle-mediated exocytosis. So essentially, the substance is simply just passing through the cell. So in conclusion, active transport is a highly important process within a cell because it involves, and this is quite important, the movement of a substance from a place of low concentration to high concentration. It can involve primary active transport, which involves the direct hydrolysis of ATP, providing energy for the process, and involves two main steps, endocytosis and exocytosis, which within themselves contain pinocytosis, phagocytosis, receptor-mediated endocytosis, and in exocytosis, vesicle-mediated exocytosis. They also both contain transcytosis because this process overall contains both of these. As I mentioned earlier, primary active transport causes the conformation change of a protein. Secondary active transport involves the establishment of another ionic gradient. Bulk transport involves the movement of many molecules at once. Phagocytosis and pinocytosis involve either a substance which is solid or a liquid moving into the cell respectively through vesicles. Receptor-mediated endocytosis involves a particular substance binding to a receptor and then moving into a cell through a vesicle. And finally, vesicle-mediated exocytosis involves a vesicle fusing with the plasma membrane in order to involve the expulsion of a particular substance out of the cell. And finally, transcytosis is simply the movement of a substance into the cell through receptor-mediated endocytosis and movement outside of the cell through vesicle-mediated exocytosis. So this is active transport in a nutshell. Thank you very much for watching.